Hi everyone, this is Veronica and today we are going to discuss a very important topic that is about gene editing in human embryo. See, what is gene editing? You all know that we have genes inside every cells of ours, and these genes ultimately decide all the characteristics and features of human body, whether from skin color to eye color to our height and everything, right? So how this gene editing is done? Like, see here, there's a DNA which contains all the information and these genes, and then we are cutting off a portion of this DNA, maybe removing a gene or inserting a new gene here. But practically it is not done with this big scissors. There are molecular scissors available, like enzymes, right? Certain proteins, enzymes which act as scissors for us, because the DNA is very small, we cannot use this scissors, right? So we'll discuss that in this lecture today. For my video updates, you can follow me on Instagram and Facebook, and for the lecture, this PDF of this lecture, it will be uploaded on my Facebook profile. So guys, if you're preparing for any government exam, our pen drive courses are also available. For extra information, additional information, you can visit our site StudyIQ.com, or you can call on these numbers. So there's one question from biotechnology, genetic engineering, which was asked in 2013 UPSC Prelims, and let's read this question. Recombinant DNA technology, genetic engineering, allows genes to be transferred across different species of plants, from animals to plants, from microorganisms to higher organisms. So you have to choose the correct answer and give the answer in the comment section. So what is the relevance of this topic? Obviously it is in news as in current affairs, so it may be asked in your any exam in MCQs if you have seen. Plus gene editing, there's an ethical dilemma associated with this topic also, so it might be asked in your GS4 ethics and integrity paper, and rest in your science and technology paper. So now why it is in news? See, recently a second international summit was held on human genome editing on 27th to 29th November this year in Hong Kong, and there a scientist, Chinese scientist, his name is Jiankui, he claimed that he has genetically manipulated twin embryo to save them from HIV infection. So he has done genetic engineering, gene editing on them. It has sent shock waves to the scientific community, because there are many strict regulations regarding when you're doing gene editing in human embryos. It is not allowed in many countries, but yet he was able to perform that, and he said that he's proud of his experiment. But let's see what are the concerns. So we'll talk about his research. His name is Jiankui, so we'll call him he. So his research, he experimented on 7 couples who volunteered to him. One of the partner among that couple, in each couple, was HIV positive, and the aim of this scientist was to prevent their child from HIV. This was all his research was based upon. And for gene editing, there is very recent technology which is being used, and this is latest technology, that is CRISPR Cas9 technology. You need to remember the name of this technology. It may be asked in your MCQs. This is used for gene editing, and this is very important, as it is right now it is the best technology available in the scientific market, right? So it is a cut and paste tool. I will explain you this technology later. So you got what his research was. And now what he did in this research, before discussing the technology, let me tell you what he did. There is a gene in us, that is CCR5, and this gene is responsible that it allows HIV virus to infect cells. Human immunodeficiency virus, you know about this. This is the gene which allows this virus to infect our cells. And what the scientist does, he disabled this gene. Okay, he totally switched off this gene so that HIV infection do not it cannot be passed to the child of the HIV positive couple. Okay, and then he used IVF. IVF is in vitro fertilization. You must have heard about the test tube babies. Okay, test tube babies, they are done through IVF. So later he implanted the embryo, the genetically modified embryo, into the womb of the mother using IVF technology. And as you know, if we have done any change in the gene, in the DNA, it will be inherited to our next generation. You need to remember that whatever changes he has done, they are inheritable. Now let's talk about the I'm telling you continuously about this technology CRISPR, so I will tell you more about this technology now, that how it is used. So for CRISPR, what is the full form? Clustered regularly interspaced short palindromic repeats. So you don't need to know more of technical term, what is palindromic repeats, you can skip that, but you should know the full form. So it is the best efficient tool in the market right now for gene editing. Okay, so let's see what is done in this technology basically. Sorry, yeah. So let's suppose this is the DNA of a human embryo. Okay, this is the DNA which contains all the information. This is the DNA which will decide how the person will look, what will be his height, skin color, what kind of diseases he is susceptible to. Everything is there. The information is in coded form in this DNA. Now we know that there is a disease causing gene here. Okay, and we want to remove this gene with the scissors, but obviously we cannot use this scissors. There are molecular scissors known as enzyme, and here the molecular scissor, the work of this scissor will be done by Cas9. This is the enzyme. So what we need to identify this place now, we will send a guide RNA which will finally go and bind here. It will identify that yes, this is the gene which can cause problem. So it will be identified. Then it will what we'll do, we'll send Cas9. This guide RNA has also, it has two functions. Firstly, it will go and bind the target gene, and next it has ability to bind this Cas9 enzyme also. Now this Cas9 will come, it will act as a scissors, and finally it will cut the DNA from here like this. Okay, so we deleted a gene here. With the CRISPR technology, Cas9 technology, we deleted this gene which was causing a certain disease. I'm just giving an example of a disease. Okay, so what we can do, when we have deleted a gene, DNA has a capability of auto repair. Okay, it can repair itself. So when it will start repairing again, we can even insert a new gene. If we want to, if we removed a small height gene, just for example, and now we want to insert a high gene of a taller height, that the person will have will be taller, we will insert a new gene when DNA is auto repairing for the part that has been deleted. So this is the technology. Now what that Chinese scientist did, it removed a gene CCR5. Okay, what is the name of the gene? CCR5, right? So CCR5, suppose it was the gene, he deleted that because this gene was responsible for HIV infection. It allows HIV virus to infect the cells, and once if it is deleted, now the infection will not pass on to the child. I hope you got this technology. I tried, because this is a cut and paste technology, right? Like you do it in your laptops, you cut something and you paste it somewhere else. This is similar to that. I hope I made you understand this. Now this technology was, it was mimicked from bacteria. Bacteria has this enzyme, originally Cas9, which it uses against virus attacks, right? See, genetics is very interesting, especially genetic engineering. Like you see in lizard, what capability it is having. If you cut its stale, it will regenerate after some time. So there is definitely a mechanism behind that. If we are able to replicate that mechanism in humans, it will be a boon. If our hand is cut in some accident or something, will be able to regenerate it. That is what scientists are continuously studying about. So this is a similar and very interesting field. Right, so here he has used Cas9 protein. This is what all what is written here. I have made you understand by using the diagram. Now just see how useful this technology has been so far. So first use is for the treatment of diseases. If there are certain genetic diseases, or we know that why cancer is infecting the cells, how we can prevent that, and many terminal illness diseases where the genes are responsible, we can treat those diseases with the help of this technology, like he did it in the embryos. This Chinese scientist, he did that the children do not get infected with HIV, but still we don't know about the precision of this technology. More study is still needed. That is why the leading scientists are in shock that how this scientist, because they have called for a global pause on this technology, at least on human embryos. They are still using this technology with plants and the lower organisms, but for humans there was a global pause because we need to study more before we can experiment it on humans, because the reason being it is inheritable, right? So what are the concerns? Now we'll discuss about the concerns. Biggest complication with this technology is to create a designer baby. Like if somebody wants that, I want the eyes of my child to be like Aishwarya Rai, so they can mimic the gene like that. I want a taller baby, so you can have designer babies. So this is the complication. So what is the complication? Like with the Chinese this experiment, I told you he removed the gene CCR5. So this gene has some other functions also. One function is negative, that is it allows the HIV virus to infect the cells, but there is a positive function also. What is the positive function? That it prevents us, the humans, from various kind of flu. But when we have finally switched off this gene completely, the child will be susceptible to higher risks of flu. So he will be there will be susceptibility of flu. So you understand. So once you have ordered, suppose you have gone for a designer baby, and then there's this cartoon showing that they have found a design fault. You have removed the CCR5 gene, but there is a certain other fault in a child. What will you do then? Right? So more research is needed on such subjects. Now about targeting. Now see, the Crispr Cas9 technique is also controversial because it is known to cause damage by off target. Off target, what does that mean? When I made you understand that Cas9 is acting like a scissors, and a scissors is a scissors. If we are targeting, suppose this is the DNA and we are targeting this part. Finally it has cut down this part, but the scissors is still there. It may go and cut a different part also. So that is off targeting. It may go and cut a useful gene. So it will damage the untargeted genes also. Since the changes are inheritable, the gene edited babies may even have to forego having children if any negative genetic traits are witnessed in the course of their lives. Right? Now population composition. It is very important because gene editing in humans with customized traits could empower eugenesis. That means in future there may be a superior race and an inferior race. So superior race will be the genetically modified babies, the children generation, and other will be the normal ones. So there will be a discrimination also and racism also, that if somebody is like, we only want white babies, we only want certain kind of babies, the designer babies we are talking, so there will be a discrimination also. So here it after some time it may be like this, that you are going to a clinic and you have to decide what kind of baby you need. So it can even come true. So these are the concerns. So let's talk about the regulation. It is banned in most of the countries. UK 14. You can perform any experiment on human embryos up to 14 weeks. 14-week embryo, then you have to discard that embryo. You cannot take it further. You cannot implant that baby, unlike the Chinese scientist has done. And that is why scientific community is in shock that they lack self-regulation, because even in China it is not allowed, but still there is no transparency that this scientist has taken this research so far that he has implanted the babies also. So now what is the ethical dilemma associated with it? See, verification. There is no course of verification, as the Chinese scientist is claiming that he has done the genetic manipulation, but tampering with the genetic material can have unintended and unknown consequences. The scientific community has no way to verify the claims on whether the gene editing was carried out in a proper manner or not. Then it's about precision, as I told you. Off targeting can happen. Approval, because in most countries of the world such experiments are banned and are punishable by law. So without regulatory approvals, there will be data and information gaps about experiments on genetic editing. And there are different consequences also, that the recent research has edited the genes of an embryo which would be passed on to offsprings and make changes in the genome of the next generation. There is a problem of designer babies. So even there are more ethical dilemmas also, like valids and unvalids. So what is this? As I told you, superior race and inferior race. Maybe in future, like today we see a lot of discrimination in the world, that there is gender discrimination, there is caste discrimination, there is racism. So it will be a new type of we are giving rise to a new type of discrimination. It is a concern. Next, this technology is very costly, so only rich will be able to afford it. Only they will help to modify their babies and get intelligent and beautiful babies maybe. So another kind of we are creating a disparity. Moreover, racism, it will give rise to racism. So such kind of ethical dilemmas, they are being talked about. And there are some more concerns, because it may be a weapon, it could be used as a weapon of mass destruction, bioterrorist. So you are creating a different generation altogether, very muscular, to fight in the wars or something like that. So anything can happen. But this is not my opinion. I'm just showing you the pros and cons. There could be some good things regarding this technology with humans also, because even when IVF was brought, many people said, scientists were against at that time also, because they said now cherry picking will be done, and certain good embryos will be used and others will be discarded, and a lot of religious beliefs are coming in between. But today we see that IVF has come upon as a boon to the world, especially for the infertile couples. So we cannot even say, maybe if this technology is very precise and no off targeting is done, maybe it will give us good results after few years. So you can write in the comment section, what do you feel about gene editing? Is it good or bad? Or what concerns we should care for, and what precautions we should take before making it legal? Because now the doors are open. If one scientist, one country has performed it, now many other scientists will also try it. We cannot stop it. We need strict regulations for it, right? So in India there is no particular law about gene editing, but however in ICMR guidelines it is prohibited for human embryos, right? We are talking about human embryos because gene editing is being done in plants and other lower organisms already. So this is about today's lecture, and the answer of this is 1, 2 and 3. This is the correct, all the statements are right. So even there is a movie about gene editing in 1997, Get a Car. You can even watch that. The people, movie makers were so smart at that time. They made a movie on this. So what I was telling you about discrimination, so they have shown this discrimination called valids and invalids. Valids are the people who are genetically modified, genetic babies, genetic engineered babies, and others are the normal population. So they were choosing only the genetically modified people for good jobs, and for the menial jobs the other population. So it is just a concern. Let's see. I hope to see your comments in the comment section, and for any queries you can contact me on social media. Thank you.